Hi team, we're now going to look at the inquiry question, how important is it for genetic material to be replicated exactly? And we'll be focusing on the dot point model of processes involved in cell replication, including but not limited to mitosis and meiosis. Now, before we can get on to mitosis, I think it's really important that we understand the difference between DNA and chromosomes in particular, and while we're at it, we'll talk about genes as well. So let's start off by drawing a very basic version of the DNA double helix. If you've watched my other video, you'll know that in the center of the DNA double helix, we have some nucleotides, and these are adenine, thymine, cytosine, and guanine, and that these nitrogen bases have complementary pairs. So C and G will always pair up, the same with A and T. And it doesn't matter where you put them, they'll always line up like this. Now, before we learn about chromosomes, you may already know that DNA is found, if we look at a cell, DNA is found in the nucleus. So if I was to look into here, I might see the double helix that we've drawn here. So when you look inside the nucleus, you won't always see this characteristic double helix shape. Instead, sometimes you'll see a chromosome. And a chromosome has this X shape. So there is a connection between this double helix and this chromosome, this X-shaped chromosome. So a chromosome is actually just condensed DNA. Instead of having this really long double helix inside of our nucleus, at some times during this cell's life, we choose to take all of that DNA and condense it up into what becomes a chromosome. Now, there's a little bit more detail that we need to understand to really understand what a chromosome is, but in very short, simple terms, a chromosome is just condensed DNA. Before we move on to understand more about this chromosome, I quickly want to cover what a gene is. Now, you know at this stage that in the middle of our DNA double helix, we've got A, T, C's and G's, and you'll find them throughout the DNA molecule. These A's, T's, C's and G's will provide instructions for a process that we'll later understand called polypeptide synthesis, where we turn these instructions, these A's, T's, C's and G's, into a protein. Now that's important because these instructions along different parts of the DNA double helix can then code for different proteins, which can lead to different traits. For example, the combinations of A's, T's, C's and G's in this section of the DNA double helix might code for something like hair color. And the next section of the DNA double helix might then code for a different trait, such as height. Now, I'm simplifying this a lot. Hair color and height are very complicated things. They're not actually due to one section of a bit of DNA. However, this is just sort of highlighting the point. A gene is a section of DNA that codes for a particular trait. Okay, so this section might code for, let's say, hair color, and this section might code for, let's say, height. Now, if you take that idea of genes on DNA over to a chromosome, you remember we talked about a chromosome as just being condensed DNA. So whereas before height, let's say, this trait that we can see on this double helix here is quite large, if we condense DNA down into a chromosome, there's actually going to be a lot more DNA inside this chromosome than what we've drawn here. So where height might be quite a large sort of section of this bit of DNA, once you condense it down, it might actually only be a very small part of a chromosome. So if height was this part, then hair color would be the bit of DNA above. So again, because we've condensed everything, this next section up here might be hair color. And then there might be another gene above that codes for a different trait, and another gene above that codes for a different trait. So as I say, there's a little bit more to this story than just DNA is condensed to form this X-shaped chromosome we had drawn before. In fact, when I want to condense my DNA, I form something called chromatin first. And the way I do that is this. So as you can see here, I've taken my double helix, and hopefully you can see that I've sort of zoomed out a little bit. So instead of seeing a double helix, I can just see a single line, which still represents the double helix, but sort of from a more zoomed out version. Now what I'm actually going to do is, instead of just sort of squishing up my DNA, instead I'm going to wrap my DNA up around these special proteins called histone proteins. And what that forms is chromatin. So chromatin is condensed DNA that's been condensed and wrapped around, the DNA has been wrapped around these histone proteins. Now if you were to look at that from a sort of more complete picture, you would then see something that looks like this, all right? Now this is a chromosome. Now I realise that's quite confusing because previously we looked at a chromosome as being an X-shaped thing. So hold on with me for a moment. This single squiggly line is a chromosome. And in fact a chromosome is just condensed chromatin. So I'm hopeful at this stage you can see that we have one chromosome, that's condensed chromatin, and we know that chromatin is just DNA wrapped around histones. In the drawing I've got here, I've actually got two chromosomes. We call this a homologous pair. Now it's actually called a homologous pair because this chromosome has the same DNA inside of it as this chromosome. So for example, if this chromosome had instructions for, let's say, hair colour, then this chromosome would also have instructions for hair colour. So why do we have two chromosomes for the same traits? Well, the reason for that is that each of these chromosomes has come from a different parent. Let's say this chromosome has come from the mother and this chromosome has come from the father. So there are two chromosomes, one from each parent. In this case we've said this one was from the mother and this one was from the father. Finally, you might be wondering to yourself, well, you said here that a chromosome is just this sort of single rod of condensed chromatin. How do we go from a chromosome that looks like this to a chromosome that looks like this? Well, if you've watched my other video on DNA replication, you'll know that we can take a single double helix and turn it into two double helices. Now this is exactly what happens in order to turn a chromosome that looks like a single rod into a chromosome that has two branches. So as we've written here, chromosomes are duplicated before mitosis. This one strand, let's say it was this was the original chromosome, this one here, would have then been duplicated to create the second strand. Chromosomes are duplicated before mitosis, which we're going to learn about next. The duplicate chromosomes are called chromatids. So now we don't call this single strand a chromosome and this one a chromosome. We now call the whole thing a chromosome, which is a bit confusing. Instead, we call each of these separate arms chromatids, and they're held together in the center by a centromere. So as you can see, one chromatid, a centromere, and a chromosome. This is a chromosome. Before it duplicated, this was a chromosome. You tend to find when we talk about chromosomes, we're talking about a chromosome when it's at this stage.